So targeted sequencing. Targeted sequencing is a method in genomics where we want to look into specific subset of DNA or RNA regions instead of either sequencing the entire genome as we did in whole genome or looked into in whole genome sequencing, or as just a second. Okay, as we did in whole genome sequencing, that is the genome. Whole exome sequencing, that is the transcriptome. Targeted sequencing, we are going to look into a specific area of interest. This is useful when, for example, researchers are interested in specific genes or regions because of prior knowledge we have that they are related to specific disease or some biological function. In general, there are two approaches related to targeted sequencing, because target enrichment, that is, it's always part of any of these protocols, try to amplify, to clone, to have more of that quantities of that target. So here we have what's called amplicon enrichment and hybridization-based enrichment as two different ways. Okay, so amplicon-based enrichments involve using PCR to amplify the region of interest. This will end up producing many copies of these specific fragments, polymerase chain reaction. Hybridization is about using probes. So it involves using some DNA probes that are complementary to the target regions. And so these probes bind to the target regions, and then some magnetic tripyrivadine beads are used to pull down and isolate the bound DNA fragments for sequencing. Targeted sequencing has lots of applications, for example, in studying diseases like cancer and population genetics, and agricultural research for studying genes in crops or livestock, and so on and so forth. With regard to the protocol, there are some differences as compared to the shotgun sequencing. I want to get your attention here. We have bacteria or fungi, and then as compared to viruses, what happens here? And the first one, the PCR amplification comes before the library preparation, as compared to library preparation, which comes before the probe enrichment. So in bacteria, the specific DNA or RNA sequences from what we would refer to as the pathogens, so bacteria or fungi in the sample. The sample could be, it's a clinical sample, so we could have blood, tissue, saliva, and so on and so forth, which contain the pathogen, and we're interested in knowing that targeted information. And so these DNA sequences from pathogens are isolated or amplified before the sequencing library. And for viruses, the library preparation happens first, and then the enrichment or selection process. Okay, this order might be due to the nature of viral genomes, because often those are RNA, they are smaller and sometimes more fragile. So targeted sequencing, we can achieve very high sequence depth. We are talking about very high numbers, like 500 times, as compared to whole genome sequencing or whole exome sequencing. And when I say 500 times or 500 folds, This means that on average, each base in the region being sequenced has been read 500 times. And this can help in detecting rare variants in these specific genes that we are looking into. Uh, as compared to whole exome sequencing, we achieve also good sequencing depth, 50 to 100 times. And sequencing whole genome is at a different level. We are here interested in the whole genome instead of specific regions. Yes, sorry. So in terms of comparison, again, between target sequencing, looking at the whole genome, the depth of coverage of the targeted panel or whole exome, we can reach up to 1,000 times. Number of samples per sequencing run, we are sort of limited here. You know, it's more efforts. Cost of sequencing reagents per sample is hundreds of dollars. Here we're talking about, you know, roughly $15 and so on and so forth. So it's really useful to have these different layers and be able to work with different technologies depending on the case. So if I'm interested in I'm looking to specific genes of interest. Why do I end up working with the whole genome or even the whole transcriptome? In terms of advantages and disadvantages, you can look into this more as a reference. But again, targeted genes, we are looking at a set of panel of genes. Generally, it's lower cost, customizable. We have good coverage as compared to the whole genome sequencing. We can there identify novel genetic causes of diseases. Okay. In terms of disadvantages, target genes does not identify genes that are not already known. Okay. So sometimes, you know, these information that could be a new knowledge for us. Who knows? Some new mutation that happens for or new variation. So we cannot capture that easily as compared to wider panel like whole exome or whole genome. As we keep looking into these and the different types of sequencing here, uh, coverage is certainly one of the fundamental concepts in genomics, and it is crucial for understanding the quality and reliability of the sequencing data. So if I want to calculate read coverage is going to be the number of reads times the length of reads. That is the total number of bases in all reads over the size of the genome. So let's say, for example, we have 1,000 base pair region. Okay. 1,000 base pair region, and that is with 10 reads. We have 10 reads. Okay. And each 100, each of these 10 reads they are of length, each of these 100 base pairs, each of these reads is made of 100 base pairs. So we have 10 reads, each one is 100 base pair, and we have 1,000 base pairs for the region. Okay, so we would have a total of how many bases sequenced. This is going to be 1,000 for the, this is going to be, let's put this in terms of, so this is, sorry, this is the, in this case, we have L, this is L, this is the length. Let me check this. Okay, sorry, this is L, yes, this is L capital. This is the length of the genomic region or segment, okay, the number of reads is n here, and the length of each read, well, let me look into this, okay. Make sure just I'm getting this right. So we would have 1,000, okay, okay, each, well, I guess I'm having some issue with my example here. So let me look into this, bear with me. So we're trying to have a sequence of that is 1,000 base pairs region with 10 reads, okay, each is 100 base pairs. We'd have a total then of 1,000 times 10 is going to end up with 10,000 bases, okay. That is the base pair region, okay. The size of the segment then, okay, okay. So we have, let's say, 10 reads, okay. Each has 100 bases, okay. So we have 1,000 base pairs, and we have 1,000 base pairs. Well, this is not gonna be correct in this case. Okay. Let me see here. Let me do this again. The example here, we're trying to work out with coverage where we have we will end up with 10,000 bases over 1,000 base pairs. So this will give us 10 times coverage, meaning each single base is covered for 10 times. Good. But again, working with these numbers, something do not match with for the time being on the formula here. Okay. Let me give it another attempt. So we're sequencing 1,000 base pairs. Good. That is, that is the region. Okay. Okay, I guess I have some typo in the example. So what I'm going to say here is that this is the size of the region or segment. Good. And then I would say I have number of reads is going to be 100. Okay. And then the length of each read is going to be 100. In this case, the coverage here would be 100 times 100 over 1,000, which will equal, I guess, I just had some typo in the notes, ended up with this number. Okay, and this is going to equal 10 times. This is how we read it. Okay, so basically the number of reads. Okay, and when I see the coverage that we are, eventually, this is very important for us, especially you want to know the accuracy, because we know that higher coverage increases the likelihood of accurately determining the correct sequence. Okay, one of the questions would be how much coverage is enough? It depends on the sequencing application. 30 times coverage might be adequate for maybe whole genome sequencing. But sometimes, maybe higher is needed, and so on and so forth. Another concept here, which is the Lander-Waterman model. Lander-Waterman model was developed to address questions about the amount of sequencing required to ensure the entire genome is adequately represented. So it's another formula here presented. Okay. sorry again. So the formula is dependent on it's using the Poisson distribution, and I can write here the formula. It's C. Oops, let me change the color. This is going to be C, which is the coverage of the genome, is going to equal what? The size of the genome, and then this is going to be times 1 minus E, that is the exponent to the power of minus N. N here is the number of reads, okay, times the average length of the reads divided by L, which is the size of the genome. Okay, so this is the formula used, or the model, to come up. I mean, this played the role, and it was pivotal in guiding early genome sequencing, especially in the Human Genome Project. Using this model, I'm sorry. Yes, sorry. So back to this. The idea is to obtain a high quality genome assembly. Of course, a certain depth of coverage is needed. And this depth is usually much greater than 1. Now, this work, I guess, came in 1988 by Linder Waterman when they developed this for shotgun sequencing, to be able to say what is the coverage there, and that is needed. And so it played a role in terms of finding the and contributing to how much coverage is needed, eventually how many reads do we need to have, and so on and so forth. Now we come to very important technology and concept, which is chromatin immunoprecipitation sequencing. That is ChIP-Seq. Another treasure hunt question here is, have you ever heard about ChIP-Seq analysis or ChIP-Seq data? Please share this with me. Okay, so ChIP-Seq here is this technology that's going to get us into looking now at the whole story in a different way. We know there's the DNA, which is packaged, right, and made compact about around some histones, and these, when we refer to the chromatin, we're looking at the DNA with these proteins. Why it matters? Because this whole DNA is compacted and packaged, right? And so these areas where this happens at specific positions may lead us at these open chromatins, because there are instructions that are going to bind at these specific positions where the histones are bound to the DNA area. At that specific area, when things are open for us, we might get to see very important part of the messages that are in the DNA that we refer to as transcription factors. Now, transcription factors or motifs, there are these parts of the DNA where transcription factors proteins are going to bind to and then give the instructions. Motifs comes usually in the upstream region before the gene. These are kind of the switch on and off for the gene expression. So think, for example, about circadian clock genes. There are these groups of circadian clock genes, and they are related to certain motifs. So there is the gene, and then before it, there's that motif. And I'm interested in knowing this area, the motif, the transcription factor binding site, and so on and so forth. Why? Because that was going to manage the regulation and gene expression. And why I give an example of circadian clock gene? Because we know through day and night is going to be a different activity. So these genes should be turned on and off, and so on and so forth. So chromatin immunoprecipitation followed by sequencing, that is ChIP-Seq data, is really powerful technique to investigate the interaction between the proteins and DNA. So now we're looking at a different sequencing for different purposes. It helps in identifying the binding sites of DNA-associated proteins, such as transcription factors or histones with specific modifications. So in terms of an overview of that ChIP-Seq process, it starts with what's called cross-linking and shearing. So the cells of interest are treated with the cross-linking agent to create covalent bonds between DNA and any protein bound to it. Next, chromatin fragmentation. So the chromatin, which is the DNA plus associated protein, is then mechanically or enzymatically is fragmented into smaller pieces. And then immunoprecipitation. Okay, immunoprecipitation is used to pull down or immunoprecipitate the protein DNA complexes from the sample. So we need to take that specific parts from the sample. So an antibody specific to the protein of interest is used, therefore. Then there is what's called reverse cross links, okay, after purification and so on and so forth, until we come to sequencing at this stage, okay. The cross links here are reversed to separate the DNA from the protein, okay. We're trying to separate the DNA from the protein. We are now interested in these areas. We want to sequence those and then apply data analysis. We can again identify where the genome transcription factors bind, which is very important for us. So this is just a visualization of what does ChIP-Seq data would look like and how it would look like. Usually we'll see peaks that we are trying to identify and recognize. And after sequencing the DNA fragments just presented that were bound to the protein of interest using the ChIP-Seq procedure, there will be a set of sequencing reads. When we map these reads back to reference genome, we will notice areas where there's a high accumulation or pile up, okay? High accumulation or pile up, because now we are going back to the reference genome. This enriched region signify potential binding sites of the proteins or the DNA. So histone code, now histone and histones we know are protein complexes around which the DNA wraps, especially in eukaryotic cells, if you remember from previous lecture. So histones, by the way, can undergo also some modifications. So there's acetylation, which is an additional of acetyl group, and this is usually associated with gene activation. There is methylation, which is addition of maybe 1, 2, or 3 methyl groups, and this can be associated with maybe either gene activation or repression. And there are other types of histone modification, phosphorylation and simulation, and other types of histone modification. Now, what is whole histone code? What is histone code? And this is a hypothesis, okay? Histone code is a hypothesis, okay? This hypothesis suggests that the histone modifications, you know, I've mentioned some examples for, like, let's say acetylation or methylation. These are types of histone modification that happens to these proteins under certain conditions. So the histone code hypothesis suggests that these histone modifications, either individually or in combination, can lead to different outcomes in terms of gene expression regulation. That is, the pattern and combination of histone modifications can create a code that is read by the cell machinery. So the different kind of, you know, these modifications may lead us to understand something and eventually be able to reflect on what is the instruction there. For example, like certain combination of histone marks might tell us a message that this is for recruiting proteins that activate transcription. Other types of combinations might relate to attracting proteins that compact the chromatin and repress transcription, and so on and so forth. Now, it is important again to know that this is a hypothesis, although it's a powerful framework for understanding the complexity and functional implication of histone modification. The idea of this, that it is a strict deterministic code, is something argued, debatable in the domain. Okay, some researchers would emphasize a more dynamic context-dependent interpretation of histone marks. So ChIP-Seq, in terms of ChIP-Seq data, as we look into the reads that would pile, there would be some background noise. So it's signal to noise ratio. This is one of the issues that might come up during the experiment and analysis. So again, did the antibody work? Did we have enough starting material, and so on and so forth? So these may lead to some differences in terms of the reads that we align to the true binding site as compared to the noise that we may end up. We usually like to have high signal to noise ratio, means we don't have lots of noise in the case of our analysis. And again, the signal in ChIP-Seq context would represent the genuine binding sites of protein of interest to the region of enrichment, as compared to the noise, which is the background level of reads. That why would it happen? Because could be non-specific binding of the antibody, random fragmentation, and some sequencing of DNA, so we end up looking at other regions or matching other regions that are not actually of interest. In terms of basic analysis, this is what I want to finish up with. Of course, reading quality, we can work with the target and the control, because we want to get the signal versus noise. Eventually, we want to look into annotations for the motifs. For example, we are interested in finding the motifs, in this case, giving the pile or sequences that we can align. And then we have something interesting here. We can build a consensus and discover the motifs, which is, again, these are very important areas. Now, this goes through a process that starts with what's called peak calling. And peak calling algorithms are going to identify regions of the genome with a significant enrichment of sequencing read. This is one thing. Then there will be extraction of peak sequences. So the DNA sequences underlying these peaks are then extracted from the reference genome. Okay. And then motif discovery, where some specialized software would be used to scan these sequences for overrepresented short DNA sequence motifs. This is a kind of overrepresentation here, okay? So we're able to, now there are different ways into how to find motifs. There's what's called position weight matrix, profile of profiling the nucleotides as different positions, and some randomized or greedy search algorithms can be used, again, to find where do we have motifs in this area. And then different tools or databases or scanning can take place into this process. So hopefully through this lecture, I know maybe personally I wouldn't say there's lots of action here, but it is I would say, a reasonable introduction and discussion of many things related to genome assembly. This is not apparently my own area of expertise in bioinformatics, but again, um, I know how important it is. I've been from years hearing about many of these technologies, ChIP-Seq, de novo genome assembly, and so on and so forth. So it's very important for us from this lecture as we finish this to understand that there are specific things. I would say we need to know that we have different technologies. Depending on the use case, we have to decide which one we need, which technology, which workflow, which pipeline. When we're talking about with genome assembly, it makes sense to look into the coverage that we will have. And again, are we looking at targeted whole exome or whole genome? Are we looking at the genome transcriptome, or and it makes so much sense now to discuss that we started discussing genome and transcriptome, because next lecture we're going to relate to this as well with RNA-seq methods and data analysis and alignment methods and so on and so forth. So I hope you enjoyed. You know now what is some quality metrics, what are some of the terms used out there, and you are stay curious about the topic, and see you next module and next week. Thank you so much.